Dear students, after the experiment, Beadle and Tatum, they observed that different enzymes in the spores, different enzymes that are involved in arginine biosynthesis pathway were absent. So they isolated different spores that were deficient in different enzymes that are involved in the synthesis or biosynthesis of arginine. Now the basic concept is that enzymes, they are proteins in nature. And proteins, they are made up of polypeptides. Which are coded by particular genes. And the enzymes, they are involved in the catalysis of synthesis of various parts of organisms. You all know that enzymes are responsible for catalyzing the synthesis pathways inside the organisms. They are also responsible for assembling the nucleic acids, proteins, carbohydrates, and lipids. Now let us see the example of arginine biosynthesis pathway. Here we have a chromosome, these are the gene clusters. What are these? These are gene clusters. Now this cluster is encoding enzyme E. This is encoding enzyme F, and this is encoding two enzymes, enzyme G and enzyme H. Now this enzyme E, it will catalyze the conversion of glutamate to ornithine. Ornithine is converted into citrulline by the action of enzyme F. Then enzyme G controls the synthesis of arginosuccinate and conversion of arginosuccinate into arginine is controlled by enzyme H. Now Beadle and Tatum, they concluded that Neurospora could not grow, the mutant Neurospora could not grow on the minimal medium because it was deficient in this enzyme, because this enzyme is involved in the synthesis of arginine or conversion of arginosuccinate into arginine. Now if this enzyme will be absent or it will be defective, there will be no conversion of arginosuccinate into arginine, and ultimately, as arginine is needed for the growth of Neurospora, it will not be able to survive even on the minimal medium. This whole evidence will support our hypothesis that a particular gene will encode for a particular polypeptide, which will give rise into proteins, particularly enzymes. And enzymes are involved in catalyzing pathways for the synthesis of various parts of the body of organisms, and any defect will result in genetic diseases. These diseases are genetic in origin and they cannot be easily treated, and nutrients, they play a very important role because a particular gene will encode for a particular enzyme. So if you damage that gene or information involved or included in that gene, ultimately it is going to affect the working and even the formation of that particular enzyme, and then the pathway which is controlled by that enzyme is also affected. If the enzyme will not be present, the pathway cannot be carried out, and ultimately there will not be formation of the particular nutrient inside the body or inside, as we are taking the example of neurospores. So since the enzyme was absent because the gene here, this gene cluster was damaged because of the X-rays, so that is why enzyme H was either it was not present, it could not be formed, or it was malfunctioned, due to which it was unable to carry out the conversion of arginosuccinate into arginine, and hence Neurospora could not grow on the minimal medium. Dear students, that is all about today's biology lecture. In the next lecture, we will discuss how DNA encodes protein structure. For your idea, I am giving you a basic concept that DNA is converted into messenger RNA, which carries out the formation of proteins. So these are two steps. The first step is known as transcription, in which the messenger RNA is formed according to the DNA template, and in the next step we have translation. The information is translated and particular protein is synthesized according to the instructions taken from the DNA. So this is the general concept, the basic concept about the formation of proteins. That is all about today. I hope you have understood well. Thank you so much for your time. Allah Hafiz.